Introduction to Histology. Al-Ma'arifah College. Objectives: understand what is histology. Realize the size of histological tissues. Know basic steps in tissue preparation and staining. Understand the importance of different stains. Identify different techniques of imaging in histology. What is histology? The name histology is derived from Greek histos, tissue. Logos, the study of. Histology, the study of tissues. What is histology? The study of the microscopic anatomy of cells and tissues. How? Examining a thin slice, section, of tissue under a light microscope or electron microscope. Enhanced by using different histological stains. Levels of body organization. Cells. The smallest independently living highly complex structure. Tissues. Functional unit, group of cells of similar function and origin, with matrix between cells. Organs. Several tissues grouped together. Systems. Several organs working for a common function. Levels of body organization. Tissues are made of cells and extracellular matrix. Intense interaction between cells and matrix. Cells and extracellular matrix form a continuum that functions together and reacts to stimuli and inhibitors together. Tissues. For fundamental tissues are recognized: epithelial tissue, connective tissue, muscular tissue, nervous tissue. The size spectrum. The small size of cells and matrix components makes histology dependent on the use of microscopes. Equal to 10⁻⁹ meters, a billionth of a meter. The size spectrum. Electron microscope. 0.1 nanometers. Equal to 10⁻⁹ meters, a billionth of a meter. Light microscope. 300 nanometers. Unaided eye. 300 micrometers. 1/1000th of a millimeter, or 0.001 millimeters, the size spectrum. Unaided eye. 300 micrometers. The size spectrum. Light microscope. 300 nanometers. Unaided eye. 300 micrometers. The size spectrum. Electron microscope. 0.1 nanometers. Light microscope. 300 nanometers. Unaided eye. 300 micrometers. Light microscopy with a maximum magnification power of x 1000 times. Electron microscopy with a maximum magnification power of x 1,000,000 times. Electron microscopy. Bronchiole, epithelial cells, x 5000. Surface of epithelial cell, x 100,000. www.visualhistology.com. Basic techniques. Preparation of histological sections. Fixation, processing, dehydration and clearing, embedding, cutting, staining, permanent mounting. Preparation of histological sections. Fixation. Fixing tissues by chemicals so they will not change their volume and shape during processing. Keeps tissue as close to their living state as possible. Prevents autolysis and bacterial attack. Prepares tissues for staining. Fixatives. Acetic acid, formaldehyde, ethanol, glutaraldehyde, methanol and picric acid. Preparation of histological sections. Dehydration and clearing. Removes fixative and water from the tissue and replace them with dehydrating fluid. Uses hydrophilic or alcohol solutions to extract water. Examples of solutions: ethanol, methanol, acetone. Preparation of histological sections. Embedding is the process by which tissues are embedded in a medium, agar, gelatin, or wax, which when solidified provides sufficient external support during sectioning. Preparation of histological sections. Cutting. Small, micro, sections are cut by a microtome. Section thickness 2 to 25 micrometers equals thick for light microscope. 60 to 100 nanometers thick for electron microscopy. Preparation of histological sections. Staining. Various stains are used to see and highlight specific structures and molecules. Preparation of histological sections. Staining. Various stains are used to see and highlight specific structures and molecules. Hematoxylin and Eosin, H&E. H&E stains are universally used for routine histological examination of tissue sections. Staining techniques. Hematoxylin and Eosin, H&E. Hematoxylin is a basic dye that stains acidic components of cells. The blue, basophilia, stains the nuclei of cells, and the of cytoplasm. Staining techniques. Hematoxylin and Eosin, H&E. Eosin is an acidic dye that stains the basic components of cells reddish pink, acidophilia. Most of cytoplasm of cells is stained by eosin. Hematoxylin and Eosin, H&E. Nuclei, blue, with some metachromasia. Cytoplasm, various shades of pink identifying different tissue components. Features of H&E. Hematoxylin Eosin. Blue colored. Positively charged, colors negatively charged molecules, such as DNA, myrrh. The ability of such anionic groups to react with a basic dye, basophilia. Eosin. Red colored. Negatively charged, colors positively charged molecules, amino acids, cytoplasm. The ability of such cationic groups to react with an acidic dye, acidophilia. Staining techniques. Periodic acid Schiff, PAS, staining. Mucus, basal lamina, glycogen, carbohydrates. H&E stain does not preserve mucus in goblet cells, which appear empty. PA stain preserves mucus in goblet cells, which stains it with a magenta color. Wheater's functional histology, a text in color atlas. Junquera and Carnero, basic histology, a text in atlas. Staining techniques. Periodic acid Schiff, PAS, staining. Mucus, basal lamina, glycogen, carbohydrates. Glycogen. Staining techniques. Periodic acid Schiff, PAS, staining. Mucus, basal lamina, glycogen, carbohydrates. Carbohydrate content in liver tissue. Stain magenta with PA stain. Clark F. Millett, University of South Carolina, USA. Staining techniques. Toluidine blue, metachromatic stain. Blue stain that stains specific components of tissues a purple color. Metachromasia is seen in the matrix of hyaline cartilage, or in the granules of mast cell. Eidvall, Omar El Safori. Science Photo Library. Blue stain of hyaline cartilage. Metachromasia of mast cells, skin. Staining technique. Soil red O Sudan black. Stain lipids red-orange in unfixed frozen sections. Sudan black. Stains lipids black in unfixed frozen sections. Staining techniques. Impregnation. Silver impregnation techniques are also widely used to demonstrate reticular fibers. Lymph node, silver impregnation. Seoul National University. Staining techniques. Reticulin. Reticular fibers are stained dark. Used especially in liver. Smooth muscle, reticulin stain. Clark F. Millett, University of South Carolina, USA. A liver biopsy stained using the reticulin demonstrating the normal hepatic plate thickness. Plasmodium falciparum, malaria. Staining techniques. GM stain. Blood smears, for disease. Wuchereria bancrofti. Causing filariasis. Plasmodium falciparum, malaria. http://parasitewonders.blogspot.com/ www.sciencebuzz.org. Plasmodium falciparum, malaria. Staining techniques. GM stain. Blood smears, for disease. Trypanosoma sp. Plasmodium falciparum, malaria. CDC/Dr. Myron G. Schultz 1970. www.sciencebuzz.org. Preparation of histological sections. Mounting. Preserves and supports a stained section. Mounted on a clear glass slide, and covered with a thin glass cover slip. Basic techniques. Total preparation time, 16 hours. Immediate frozen section time, 5 minutes. For immediate results, in tumor surgery. Results not as accurate as formalin-fixed, wax-embedded specimens. Basic techniques. In some cases the tissue to be examined is a very thin membrane, cell smears. Blood or bone marrow. Basic techniques. In some cases the tissue to be examined is a very thin membrane, cell smears. Blood or bone marrow. http://hepatitisresearchandnewsupdates.blogspot.com. Basic techniques. In some cases the tissue to be examined is a very thin membrane, cell smears. Blood or bone marrow. Epithelial cells. For example from the oral cavity, cervix uteri. Cervical carcinoma, Pap stain. Unit of Oklahoma Health Science Center. National Cancer Institute/Science Photo Library. Some definitions: histology, biopsy, autopsy. Microscopic study of tissues. Biopsy. Removal of tissues for diagnostic purposes. Autopsy. Examination of organs of a dead body, to find clues to determine cause of death. Phase contrast microscopy. Phase contrast microscopy. An optical microscopy illumination technique. A small phase shifts in the light passing through a transparent specimen are converted into amplitude or contrast changes in the image. Does not require staining to view the slide. Epithelial cell from cheek. Polarizing light microscopy. Polarization is a property of electromagnetic waves, such as light, that describes the orientation of their oscillations. Polarizer is rotated to transmit the reflections as well as possible. By rotating the polarizer by 90 degrees, almost all reflected sunlight is blocked. Polarizing light microscopy. Polarization is a property of electromagnetic waves, such as light, that describes the orientation of their oscillations. Polarizing light microscopy. Collagen fibers. Yellow birefringence. Fluorescence microscopy. A sample is illuminated with light of a one wavelength which causes fluorescence in the sample. The light emitted by fluorescence, which is at a different, longer, wavelength than the illumination, is then detected through a microscope objective. Fluorescence microscopy. Epifluorescent imaging of the three components in a dividing human cancer cell. DNA is stained blue. A protein called INSEMP, intercentromere protein, is green, and the microtubules are red. Each fluorophore is imaged separately using different filters, and images are captured sequentially using a digital CCD camera charge-coupled device, then overlaid to give a complete image. Fluorescence microscopy. Scanning electron microscopy. Clark F. Millett, University of South Carolina, USA. Scanning electron microscopy. Bruce Wetzel/Harry Schaefer, courtesy National Cancer Institute. Scanning electron microscopy. Scanning electron microscopy. Scanning electron microscopy. Cutting of human hair. Catchendum.blogspot. Scanning electron microscopy. Human skin. Summary. What is histology? The size spectrum and organizational structure. Light microscope, electron microscope. Summary. Preparation of tissue, different stains and special techniques. Fixation. Processing. embedding, cutting, staining, mounting. Different stains and special techniques.